The International Genetically Engineered Machine, iGEM, competition, founded by MIT, is an international academic competition centered on synthetic biology. It integrates engineering, business, information science, and humanities, aiming for participants to address social issues. Teams develop marketable, environmentally friendly products and engage in practical and local community activities, enhancing their social responsibilities. Each team selects its research topic and refines their ideas throughout the competition process.
Team divisions. Teams are divided into high school and university categories. Research topics. Each team provides its own research topic and refines its ideas during the competition. Subdivisions within teams. Teams are further divided into 3 groups. Wet lab focuses on experimental work involving biological materials. Dry lab involves computational modeling and data analysis. Human practices explores the ethical, legal, and social implications of the project. These groups work interdependently, ensuring a comprehensive approach to their project, and collaboratively complete the competition requirements.
Wet lab. Responsible for reading research papers, determining principles, formulating experimental topics, and refining experimental procedures. Dry lab. Focuses on creating physical models and writing programming code, involving fewer chemical reagents. This group typically complements the work of the wet lab, developing differently according to the specific topic. Human practices. Handle social outreach, event organization, fundraising, and other non-laboratory activities.
This stage is critical for our success and involves key phases such as registration, promotion, recruitment, idea discussion, knowledge learning, and work planning. Stage 0, June 2024, January 2025. 1. Registration and promotion, June, August 2024. Focus on signing up and spreading the word about our participation. 2. Preliminary preparations, September 2024, January 2025. Team recruitment. We will actively recruit team members who are passionate about synthetic biology and willing to dedicate their time and efforts to the competition. Idea discussion. Team will brainstorm and lead the project by discussing and selecting innovative ideas. This phase will drive our overall process. Knowledge learning. We will engage in extensive learning to acquire the necessary knowledge related to our project theme. This includes reading research papers, studying relevant principles, and gaining expertise in techniques. Work planning. Finally, we will develop and plan the specific tasks required for our project. This involves creating detailed schedules, assigning responsibilities, and ensuring that all aspects of the project are well coordinated. Phase 1, February to May. Team formation. Identify the final team members from interested students. Project idea discussions. Ideally, the iGEM project should be entirely student-driven. Teams often hold multiple brainstorming sessions, sometimes with mentors and sometimes without. Setting team goals and tasks can be challenging, but it's worth the effort. Listing high-level goals and detailed tasks needed to achieve them will help your team stay focused. Reviewing competition requirements. Make sure your team understands all the requirements, deliverables, and deadlines. This is a team responsibility, not just for one member. Phase 2, June to August. Starting project work. Begin your project by getting protocol training, adhering to school safety rules, and following iGEM safety policies. Building your design. Order your DNA, and assemble your genetic devices and systems. Conducting functional experiments. Once you have functional devices, proceed application, testing, and data collection. Processing and plotting results. Determine the software needed for data processing. Decide on display units, include controls, and accurately present your data in charts, graphs. Working on your team wiki and registry pages. Consistent project documentation is crucial. Continuously update wiki and registry pages throughout the project. Don't leave it until the last minute. Phase 3, September to October. Presenting your project materials. Once your final data is plotted and ready, create materials to present your project at the Jamboree. Completing your project documentation. Finish your documentation, including the team wiki, registry part pages, and judging form. Preparing and practicing your presentation like your wiki. Work on your presentation throughout the project. Practice in front of an audience to hone your speaking skills, and incorporate feedback to refine your content. Attending the Grand Jamboree in Paris, meet other iGEM teams, celebrate your work, and have fun. iGEM possible topics: climate crisis, atmospheric greenhouse gas reduction, sequestration, oceanic carbon sequestering, carbon fixation, renewable energy. Environment, local environmental pollution, ocean pollution, clean drinking water, antimicrobial resistance, algal blooms. Conservation, biodiversity, social insect health, antimicrobial resistance, coral reef protection and restoration, invasive species control. Food and nutrition, detecting food spoilage, nutritional deficiencies, sustainable aquaculture, non-animal protein, packaging and storage of food. Diagnostic, CRISPR, Cas9, vector-borne diseases, targeting the vector, public health and pandemic preparedness, detecting nutritional deficiencies, microfluidic devices. Therapeutics, ERNI, CRISPR viral vectors, biofilms, gene therapy, vaccines, probiotics and nutritional supplements, fertility, contraceptives, uterine health, UV, radiation prevention. Foundational advance, novel chassis, advancements in DNA assembly, DNA data storage, biosafety concerns, biocontainment, release, dual use, new hardware tools. Biomanufacturing, scale manufacturing, fabrics, tissue engineering, e.g., new skin, organs, fragrances, clean dyes and pigments, sustainable mining. Software and AI bioinformatics tools, predictive design, modeling tools, design tools. BR remediation, plastics and microplastics, polycyclic aromatic hydrocarbons, PAHs, pesticides and agrochemicals, heavy metals, household and industrial waste, toxic waste from nuclear facilities or contaminated sites. Agriculture, biological fertilizers, microbial nitrogen fixation, crop yields, stress tolerance, diseases and resistance, soil health. Space, in situ resource utilization, space manufacturing, life support, space medicine, space cybernetics, terraforming. This slide outlines various potential topics for iGEM projects, ranging from climate crisis solutions and environmental protection to advanced diagnostics, therapeutics, and space exploration. Each topic presents opportunities for innovative research and development within the competition.